It is really my honor to be here and welcome Dr. Tomaselli, who is the Dean of the Albert Einstein College of Medicine and for several decades had a critically important role in establishing work at Johns Hopkins with multiple partners in the field of electrophysiology, particularly on the focus on sudden cardiac death, which he's going to be talking to us. As a graduate of Albert Einstein and someone who was a few years after him, I'm really honored to ask Dr. Ricard to do a formal introduction and really would like to welcome Dr. Tomaselli here with us today. So, Joe?
Thanks, Eric. It really is a pleasure to have you with us, Gordon. Dr. Tomaselli, as Eric mentioned, is one of the biggest leaders in the field of EP. His medical journey started, as Dr. Velasquez mentioned, in Albert Einstein, where he did medical school. He subsequently went to UCSF for residency and then did his cardiology fellowship at Johns Hopkins. And he stayed at Johns Hopkins for a while, and he very quickly rose through the ranks to a full professor. Along the way, he was the director of the arrhythmia service. He was the director of molecular and cellular cardiology, and ultimately became the chief of cardiology with the Michelle Morawski Endowed Chair, and did that for well over a decade. After which, a year ago, he basically did a complete 360 and went back to where it all started, which is Albert Einstein, but now in the capacity of dean, as a senior vice president and chief academic officer for Montefiore Health. He is extremely well published. I can't even begin to go into his record in terms of peer-reviewed publications, more than 250 peer-reviewed publications, other than the editorials and book chapters and reviews and all that. And what is very interesting is that I was looking at your CV, and one of his very early publications, among the earliest publications that he's had, was a senior authorship in JCI, and he's done another 360 where he's come back to JCI, but this time around as an editor-in-chief a couple of years ago. He has multiple patents, I think 5 or 6 patents. He has mentored literally a generation of investigators who are now leaders in the field of basic and translational EP. He has been honored very numerous ways, including being the president of the American Heart Association and receiving multiple other honors, including the Gold Heart honor at the AHA. And on top of the basic science experimental work, on top of the educational work that he does, he continues to see patients, which is really unbelievable. He truly represents the triple threat, and we are really honored to have you here with us, and we look forward to your presentation.
Thank you. So it's a delight for me to be here, and thank you, Eric. Thank you, Joe, for those kind words. It's been a fun ride for me. I spent enough time at Johns Hopkins to have 3 kids and a grandchild there. 1 additional grandchild in New York, but it's been for me a great run. And what I thought I'd do today is talk about what we've been doing continuously for the last 2.5 decades in collaboration with a number of other investigators at Johns Hopkins primarily. This is something that I thought would be good for kind of a general audience. And what I'd like to do is talk a little bit about electrical remodeling that occurs in heart failure and how you might be able to reverse that electrical remodeling simply by a mechanical therapy, CRT. So the objectives today are really to describe the features of the electrical changes that occur at the cellular, molecular level, how they might contribute to arrhythmogenesis in the failing heart, and how CRT might reverse some of these changes. And in particular, I'll focus on ionic currents, calcium handling, and some of the changes that occur in cell metabolism in a failing heart. Then I'd like to finish, since I know this is probably a mixed audience, talking a little bit about a study that we started now 15 years ago. It's a prospective observational study of patients who've gotten primary prevention defibrillators, and how that therapy might have been related to and informed by some of the more basic work that we were doing. So this slide reminds me to tell you two things. One, we really don't even have a good handle on the epidemiology of sudden cardiac death. And if one uses death certificate adjudication, you overestimate the incidence of sudden cardiac death, probably by about twofold. The better data are data that come from prospective, multi-source types of studies like the Oregon SUD study. And I think this is probably close to being a real number for sudden cardiac death, about 200,000 cases per year in this country. Now, the other thing that's important to recognize about sudden cardiac death, like virtually all forms of heart disease, is that coronary artery disease is lurking somewhere around, contributing to the incidence of sudden cardiac death. So if you're doing any studies, omics studies, functional studies of heart failure, you're probably also at some level in humans studying coronary artery disease as well. And as such, as a disease of aging, age has an important influence on not only remodeling, but the effects of heart failure in humans. So I probably don't need to remind this audience about the other kind of major epidemiologic part of this talk, and that's heart failure and the scope of the problem. These are data from the American Heart Association update in 2018. That's when I left Hopkins. I probably need to update this again at some point soon. But suffice it to say, highly prevalent, lots of new diagnoses. Basically, more than a million hospitalizations a year, an incredible cost in terms of direct cost in terms of hospitalization, but also cost in terms of productive years of work lost. 75,000 deaths per year, and in fact, the incidence of sudden cardiac death increases dramatically in the presence of heart failure in the Framingham Heart Study, about a 6-fold increase. And in fact, this is an equal opportunity malady, killing as many women as it does men. There's about a 50% incidence of sudden cardiac death. Now, whether or not that's true, arrhythmic sudden cardiac death is debatable. We have a number of pharmacologic treatments for heart failure, as you're well aware, neprilysin inhibitors, beta-adrenergic receptor agonists, all of which can actually improve contractility and, in fact, improve symptoms, but improving contractility sometimes comes with the cost of increasing mortality. CRT is relatively unique in the sense that, and I'll come back to this in just a minute, it also can improve cardiac performance, but does so with more efficiency in kind of energetic performance. So now, many years ago, we studied the fundamental changes that occur in a model of heart failure. And the model of heart failure that we use is rapid ventricular pacing, first in dogs and in pigs. But I would say that there are two things about this model of heart failure. One, tachypacing-induced heart failure is a bit unique, and there's probably not a clinical form of this. But I think it does a reasonable job at representing what occurs in dilated cardiomyopathy. But there are two things about rapid ventricular pacing that I think you need to keep in mind. The tachycardia piece of it does produce heart failure, but the pacing piece of it also produces desynchronous contraction. So it's really a model of both heart failure and desynchronous contraction. And we've used several iterations of this particular model. The basal implementation is 3 to 4 weeks of pacing at between 200 and 240 beats per minute from the right ventricular apex. And reliably, after about 3 weeks, LVEDP rises to about 35 to 40 millimeters of mercury. This is a representation of what happens electrically. These are action potentials that have been recorded from cells isolated from different regions of the left ventricular free wall. And two things are apparent. One, action potential duration prolongs. So repolarization reserve actually goes down. The other thing that happens is that if you look at the difference between the longest and the shortest action potential, the dispersion, and there is normally physiologic dispersion of action potential duration in the heart, with the epicardium being a little bit shorter than the endocardium, and in fact, that allows activation to proceed endocardial to epicardially, and then recovery to proceed epicardially to endocardially. Hence, the morphology and the polarity of the QRS complex and the T wave normally are the same direction because the activation and recovery are in different sequences. But suffice it to say, a couple of things happen. Action potential durations prolong. This gets particularly exaggerated at slow rates. And the delta APD, difference between longest and shortest action potential duration, also are prolonged in a manner that's rate dependent. So what are the underlying changes? This looks in many ways like there's a class 3 antiarrhythmic drug on board. It also looks like an acquired form of long QT syndrome. However, there are other changes that make it even worse than an acquired form of long QT syndrome. So two essential features that contribute substantially to the changes in electrophysiology in the failing heart. One, this reduced repolarizing reserve action potential prolongation, enhanced dispersion of repolarization, but there are also changes in the way the cell handles calcium. So in various models, and there have been many models of heart failure that have been studied in electrical remodeling in these models of heart failure, I would say that one of the consistent findings is that virtually every repolarizing potassium current in the heart in one or more of these models has been demonstrated to be downregulated. Similarly, in the model that we use, we see downregulation of virtually every repolarizing potassium current, the transient outward current, which controls the early part of the action potential, and the delayed rectifier components, which are the channels that are mutated in long QT syndrome, and those that are blocked by drugs with class 3 antiarrhythmic action are downregulated as well. The channel that governs the resting membrane potential, the inward rectifier potassium channel, also is downregulated. So reduced repolarizing reserve, action potential duration prolongation, but the other thing that happens is the cells don't handle calcium in a normal way. These are Indo-1 calcium transients, so it's really a measure of the intracellular calcium during the cardiac cycle. In green is normal, so calcium after activation rises rapidly and then decays quite rapidly as well, getting back to basal levels of calcium. In comparison, the calcium transient that occurs in a failing heart cell is very much reduced in amplitude, and there's a slow creep in the level of calcium which lasts for as long as the action potential lasts. The kinetics are also really quite slow. And we'll talk a little bit about why that happens, but in fact, getting back to normal levels of resting diastolic calcium really takes quite a long period of time. We looked and did this with Brian O'Rourke many years ago by serially inhibiting SERCA and the sodium calcium exchanger. These are two of the mechanisms by which cytosolic calcium gets reduced back to normal levels during diastole. SERCA by reuptake of calcium into the SR, sodium calcium exchange, and electrogen exchange of calcium for sodium with extrusion from the cell. The short story is that the normal cell depends, the non-failing cell depends more on SERCA for restoration of normal intracellular calcium levels, whereas the failing cell relies more on the sodium calcium exchanger. The implications of that are that SR loading is dramatically altered in the failing heart cell and reduced. And that may be one of the reasons why you have a flat or even a negative force-frequency relationship in heart failure compared to in normal hearts where, in fact, increasing rates of stimulation result in increasing contractility. So again, this looks like kind of long QT. This looks like something else. The crosstalk between the two is particularly bad in terms of producing changes in arrhythmia susceptibility. Again, this certainly influences excitation-contraction coupling, but there are profound effects on the electrophysiology as a consequence of altered calcium handling in this model. So we have to be not so myopically myocentric here. Arrhythmias are not cellular phenomena, but they're tissue phenomena. And this is work done by one of the best postdocs I've ever had in my laboratory. That's Fadi Akar, Joe's brother, who worked in my lab for 4 or 5 years. And what he was interested in is what are the network properties that are altered in the failing heart. And he used a number of different techniques, but in this case, he used perfused myocardial wedges from the canine that were loaded with a voltage-sensitive dye and were pasted at the sites labeled S1. Now, what you can see is that in the presence of heart failure, and these are isochrone lines, the isochrone lines get more crowded, so conduction velocity is slower. Yet, it still remains anisotropic with the conduction velocity along the fiber axis being faster than across the fiber axis. But suffice it to say, conduction velocity was profoundly diminished in in failing hearts compared to control hearts. So now we have three things that have gone wrong. The active membrane properties of the cell are altered, calcium handling is altered, and then the network properties, conduction velocity is altered. So this is if one thinks about wavelength properties of cardiac tissue, this is a disaster waiting to happen. So the susceptibility to arrhythmias is really quite high. So conduction velocity decreases both when viewed from the epicardial and the endocardial surface. The etiology probably has a lot to do with, since this model of pacing tachycardia produces very little fibrosis in a matter of 3 to 4 weeks, but instead there are alterations in the expression, post-translational modification, and localization of the major intercellular ion channel, connexin 43. Connexin 43 in normal heart cells is very well behaved. It lives mostly in the intercalated disc between cell-cell junctions. Three things happen to connexin 43. The total amount of immunoreactive protein goes down in heart failure. The other thing that happens in this bit of a gained up view here is that there's more connexin 43 along the lateral border of cells, so so-called lateralization of connexin 43. But keep in mind that there's no change in the anisotropy of the heart, so conduction velocity doesn't change, but there's more protein along the lateral border. And the other thing that changes is when we look at a hypophosphorylated form, less active form of connexin 43 in heart failure, despite the fact that total immunoreactive protein goes down, this hypophosphorylated form goes up. So less function, more of this hypophosphorylated form of connexin 43. Now, if we look at cell-cell borders, one cell, two cells, and we look at the lateral border here, we see these pentalaminar circular membranes. These are annular gap junctions. These annular gap junctions, if we stain with immunogold labeled for connexin 43, are just chock full of connexin 43. So we have a lot of protein between cells, but that protein doesn't exist as it should with connexin 43 with two hemichannels and adjacent cells. Instead, they're in a recycling membrane in between those two cells. So that explains why there's more protein, yet there's no change in the transverse conduction velocity, in fact, a decrease in transverse conduction velocity in the failing heart. So that's the baseline change that occurs in heart failure. We got interested, and this was a part of a SCORE grant with Dave Cass, Brian O'Rourke, and others, interested in looking at what happens to a failing heart when you kind of resynchronize it. And this is work that was done by Greg Nelson in Dave's lab now many years ago. These are human hearts, and he looked at using MRI the pattern of contraction here. And one can see, if the movie plays, let's see. All right. The green dot shows you where the septum is. The septum's activated early. When it's activated early, it contracts and pushes the lateral wall out, so more stress in the lateral wall. By the time that activation wave gets around to the lateral wall, it contracts against a pre-contracted septum. That means that the stress in that lateral wall, particularly in the endocardium, is really quite high. And that's to be contrasted with what happens in the normal heart. In the normal heart, there's very orderly activation from endocardium to epicardium without dysynchrony within the chamber itself. The question is, what happens when you resynchronize contraction of a failing heart? Now, there are many ways to improve contractility, and you can do so pharmacologically with beta-adrenergic agonists. But if you do that, you do that at the price of, with dPdt being an index of contractility, you do it at the price of increasing myocardial oxygen consumption. CRT is absolutely unique in the sense that you can get the same amount of improvement in contractility, but you do so at a lower energetic cost. So you've improved the efficiency of performance, at least with respect to myocardial oxygen consumption, in the failing heart if you resynchronize contraction. So again, I want to remind you that the model that we study is heart failure, but heart failure with dysynchronous contraction. And what we've done is we've implemented a few variants of this model to study specifically what CRT does, but it's really not CRT. Let me qualify exactly what we're doing here. So in the next iteration of this model, we created, with catheter ablation, left bundle branch block, and then used right atrial pacing at 200 beats per minute for 3 weeks. This is kind of baseline. Then we divided the animals into two groups. One group where we continued right atrial pacing at 200 beats per minute for an additional 3 weeks. And we call this dysynchronous heart failure, DHF. The other group of animals, we put an epicardial lead in the left ventricle, and we resynchronized pacing. So it's resynchronized tachypacing. It's not CRT, it's resynchronized tachypacing, but I will use CRT as shorthand, and I apologize because it's really not CRT. And then we did all the same things that we did with the baseline model. So these are echo strain maps from the lateral wall in dotted lines, the septum in the solid line. And what you can see in heart failure is what we expect to see. That is, there's stretch early in systole, and then contraction late in systole. And it's dysynchronous between the lateral wall and the septal wall. And with CRT, we can absolutely resynchronize the strain that occurs in lateral and septal wall. And that's reflected also in the shortening of the QRS duration with biventricular pacing. Now, there are several things that happen between week 3 and week 6. There is a statistically significant, but probably not clinically significant change in ejection fraction. That is, if you continue to pace in a desynchronous fashion, ejection fraction continues to go down modestly. It's already bad, but it goes down a little bit more. With CRT, again, with tachypacing, ejection fraction goes up modestly, statistically significant, but probably not clinically significantly different. If you look at the cells, there's an increased level of apoptosis or programmed cell death that occurs throughout the ventricle in heart failure, which is reduced in CRT. And there are regional changes in the expression of stress hormones, particularly CAM kinase 2 and P38, and I'll come back to that in just a minute. So we have both global and regional changes in kind of the molecular functioning of hearts after CRT compared to DHF. So one of the things that we looked at with John Bridge and Frank Sachs was we said, okay, is there a change in cellular structure that might help to explain? Particularly, we're interested in the changes in calcium handling. And what we did here was we stained T-tubules with weak germ agglutinin and stained junctional SR with an antibody for the ryanodine receptor. And with a bunch of image processing, we looked at 3D reconstructions after deconvolution of the T-tubular membranes, which are shown in blue, and the red dots represent junctional SR in normal hearts, the lateral wall of a failing heart, and in CRT. And what you can see is that in the failing heart, in this kind of bread loaf slice and longitudinal slice, there's effacement of a lot of the T-tubules. And that effacement of a lot of those T-tubules means that there's not as direct a communication between the junctional SR and the T-tubule, or the dyad, which is where all the calcium handling, the business end of calcium handling actually occurs in a heart cell. And what we've focused on is this particular region here. This dyad pulls into proximity the trigger calcium that comes in through L-type calcium channels, and the calcium and the ryanodine receptor, which actually responds to this triggered calcium with an amplification of calcium release that's important for contraction. And in fact, if one looks at this, there are a number of things that happen. So the number of T-tubules goes down. There are changes as a result of this because of changes in the proximity of the L-type calcium channel and the ryanodine receptor that produce a late current, a late calcium current. That late calcium current results in action potential prolongation, a reduction in SR calcium, and an increase in cytosolic calcium. This increase in cytosolic calcium also predisposes to the development of another arrhythmia mechanism, calcium waves. So this is a model or a disease of myocardium that is really quite prone to developing arrhythmias for a number of different reasons. So CRT seems to repair some of the cellular structure. Are there functional changes that occur in CRT that might be antiarrhythmic? And I probably wouldn't be telling you this if there weren't. But one of the things that actually does happen in the presence of CRT is that action potential prolongation, particularly in the lateral wall where it's profound, decreases. And that creates a reduction in the dispersion of repolarization throughout the ventricle. And that's shown in these plots over a range of pacing cycle lengths, where in fact there's a statistically significant reduction in action potential duration in the lateral wall, not so much in the anterior wall, but what it does is it homogenizes repolarization. So that exaggerated dispersion of repolarization seems to be a little bit less prominent in the presence of CRT compared to desynchronous heart failure. Well, why does that occur? There are a number of changes that occur in the expression of both ionic currents as well as calcium handling proteins, and the changes are relatively specific. There was a downregulation that I told you about of the inward rectifier potassium current, which governs the resting membrane potential of the cell. That actually is partially reversed in the presence of CRT. The delayed rectifier potassium currents, those that are mutated in long QT syndromes, types 1 and 2, and those that are the receptors for drugs like sotalol, amiodarone, and the like, are also partially corrected by CRT. The transient outward potassium current, the transient outward potassium current is also profoundly downregulated in heart failure. But it doesn't change. So there's relatively specific reverse remodeling that occurs with respect to potassium currents in the failing heart. And all of this is post-transcriptional and post-translational because there doesn't appear to be much change in either message levels when comparing to synchronous heart failure compared to control, or in protein levels. So it looks like it's all kind of post-translational change in function that occurs in the failing heart. The other thing that happens to one of my favorite molecules, the voltage-dependent sodium channel, is there's an increase in the so-called late sodium current. Late sodium current is the sodium current that actually is prominent in long QT type 3. And in fact, in heart failure, there's a dramatic increase. And although this looks like a relatively small amount of current, a relatively small amount of current over the entire action potential duration produces a lot of depolarizing current, and a lot of that depolarizing current, which is ranolazine sensitive, so therefore sodium channel mediated, is partially reversed simply by resynchronizing contraction. Well, why might that occur? Well, there are a number of ways that sodium currents might be modulated by heart failure and by calcium in particular. And we've, a little bit of a digression here for a couple minutes, we've been very interested in calcium regulation of sodium currents and spent the last few years of my time at Hopkins looking at this. But one of the ways that actually the sodium currents are regulated is by phosphorylation by a stress kinase, and the stress kinase is CAM kinase 2. CAM kinase 2 actually goes up pretty dramatically in heart failure, but more importantly, not only does CAM kinase total go up, but the phosphorylated form, the activated form of CAM kinase actually goes up quite dramatically in heart failure, and it's reduced back to control levels with CRT. So there's at least one mechanism by which calcium regulation of sodium currents to produce an arrhythmogenic late current might occur, and that's with upregulation by CAM kinase phosphorylation. We've been very interested in direct calcium regulation of sodium currents, and we've, along with a number of other labs, crystallized the carboxy terminus of the sodium channel, shown here, where there's an EF hand and there's an apopathic IQ motif that actually binds the most ubiquitous calcium sensor in nature, calmodulin. And in fact, calmodulin has a number of effects on the current. Binding of calmodulin to the channel increases the probability that that channel is going to open. It also changes the gating kinetics of the current. And this C-terminus, where calmodulin binds, has a number of interesting side chains that, when mutated, produce in blue Brugada syndrome, in gray long QT syndrome. So you have very different phenotypes with mutations in the same region of the channel. And in teal is calmodulin, and it's bound to this IQ motif. The C lobe is bound to this so-called H6 long helix, and the N lobe actually sits up here near the EF hand of the C-terminus of this. Now, there are a number of syndromes that are associated with mutations in the C-terminus of the sodium channel, including Brugada long QT, sudden infant death syndrome, some forms of heritable atrial fibrillation. So we were interested in trying to figure out how this all works, and another talented kind of postdoc in my lab, Manu Benjani, actually did a clever set of experiments looking at the level of sodium current with instantaneous changes in calcium. So we loaded cells with calcium that were chelated by DM nitrogen. Flash of light instantaneously changed the level of calcium from nanomolar concentrations to about 10 micromolar. That 10 micromolar change in the control circumstance reduces the sodium current, and this is something that we call calcium-dependent inactivation. It's very robust in calcium channels. Nobody really recognized it in sodium channels, but in fact there is calcium-dependent inactivation of sodium currents as well. The other thing that calmodulin does is it increases the probability that the channel is open. These are single-channel recordings of sodium currents that right after the voltage step, they open up rapidly, close kind of rapidly. and in the presence of calmodulin, they open up robustly. If you now displace calmodulin, and in fact, Greg Pitt's lab and others have shown a termolecular structure of calmodulin, the sodium channel C terminus, and a molecule that we know is increased in heart failure, fibroblast growth factor, homology factors. These are molecules that regulate sodium channels. They are non-secreted forms of FGF. These non-secreted forms of FGF don't have many ligands. The one that we know of is the sodium current, but this goes up in heart failure. And when you solve the structure in the presence of FHF1B, what happens is the situs of the calmodulin binding changes dramatically. So you still bind to this long helix, but the N terminus is no longer tucked up underneath the EF hand, but instead is flapping out in the breeze. That's associated with a decrease in the open probability. It's also associated with the absence of a change in conduction in the presence of calcium, so the absence of calcium-dependent inactivation. So a little bit of a digression and may explain some of the things that happen in more inherited forms of heart disease. So what does CRT then do to calcium handling? Well, these again are calcium transients measured with INDO1 in the anterior and the lateral wall in the control circumstance. In DHF, you can see that the transient, as I mentioned before, dramatically depressed, kinetics are slowed. CRT in and of itself homogenizes the transients. This may be a consequence to some extent of homogenization of the action potential morphology, but in fact also hastens the kinetics of restoration of normal intracellular calcium levels. This is shown in summary format here, a downregulation in the peak, which is reversed by CRT, slowing of the decay, which is also reversed by CRT. Well, does this have consequences for susceptibility to arrhythmias? Well, we used a cellular index of arrhythmias in this case, and that's so-called early after depolarizations. Early after depolarizations are interruptions in the normal process of repolarization of the action potential that occurs and are potentially arrhythmogenic because if they reach a sufficient amplitude, they can beget another action potential, a so-called triggered action potential. And one of the things that CRT does is shorten the action potential duration and eliminate a lot of the EADs. So action potential durations at various stages of repolarization are shortened by CRT, and they're shortened by CRT in both the anterior and the lateral wall early after depolarizations. Potentially arrhythmogenic changes in the action potential are also reduced by CRT. So the other thing that is a target for therapeutics and is wildly deranged in the failing heart is beta-adrenergic receptor signaling. So we looked at several different downstream effects of beta-adrenergic receptor signaling, including the calcium transient, calcium currents, as well as cell shortening. And these are cell shortening traces, and let me just, in cells that are isolated from a control heart, in the absence in green and presence of isopaternyl in a DHF heart cell, and in CRT. And what you can see is there's depression in shortening in the basal state, absence of isopaternyl. In saturating concentrations of isopaternyl in DHF, very little change in cell shortening. In CRT, cell shortening is completely restored to normal. And this is an effect that's seen prominently in both the anterior and the lateral wall and is associated with, as I mentioned previously, a change simultaneously in calcium transient, both amplitude and kinetics. This appears to be an effect that's both beta-1 and beta-2 mediated, and the mechanism that I won't have time to go into in detail is interesting. The process of beta-adrenergic receptor signaling, and in particular turning it off, is really a two-step process where the receptor is phosphorylated by a kinase, these GRKs, which actually go up in heart failure, but they also are high in CRT, so there's no difference there. The other thing that happens is the maximally stimulated activity of adenylyl cyclase by forskolin is depressed in heart failure, but it's also depressed in CRT. One of the things that does happen is there's a change in the amount of alpha I, G-alpha I inhibition. This is an inhibitory alpha subunit, G protein subunit, which actually specifically goes up in CRT. And one of the reasons that inhibition goes up in CRT probably has to do with an increase in isoform-specific regulators of G protein signaling. These are basically GTP accelerants. These GTP accelerants have very specific targets. inhibitory, stimulatory, and GQ proteins. And in fact, the ones that are specific for GI go up substantially. That's regulatory G protein signaling. Proteins 2 and 3 are specifically increased in CRT. So this may be one of the reasons that this beta-adrenergic receptor signaling is restored in this case. So let me just change gears for just a minute. We were interested now many years ago in trying to figure out what are the drivers for all this change. And we embarked upon, before in fact there was a canine-specific microarray. We created a canine-specific microarray and started to try and understand what the changes in the transcriptome were at various periods of time during the development of heart failure in this model. And we created, by looking at sequential changes in gene expression and trying to correlate that with some physiologic function, things that we call these basically functionally associated gene expression networks. And in this case, this is the action potential duration centered gene association network. So what we did was we correlated changes in the transcriptome, changes in mRNA expression, with a specific change in function, in this case, action potential duration. And what we asked was, what are the changes in the transcripts that actually mimic the physiologic changes in action potential duration during the development of heart failure? So we asked what changes occur in mRNAs that occur quite robustly between day 0 and day 3, less robustly between day 3 and day 7, even more modestly between day 7 and 14 days, and then are flat afterwards. So we asked which genes change with this pattern. We didn't care whether it was up or down regulated, but we clustered all the genes that changed in this pattern. That is, big change early, less of a change later in the pacing process. And this is a matrix plot of 125 genes versus 125 genes. And we looked at correlations between expression in this matrix and found that there were a number of genes that change in a positively correlated fashion here in orange. That is, one gene went up, the other gene went up. In a negative fashion, one gene went down, the other gene went up. That's in blue. And there were a bunch that weren't correlated. And if we ask the question, what are the genes in module 1, which is the largest module, that seem to change in a pattern reminiscent of the change in action potential duration? It turned out that of those genes, 25 of 31 genes were involved in energy generation, oxidative phosphorylation, TCA cycle, mitochondrial protein translation, and they're shown diagrammatically here in orange, the ones that changed. So then we asked, okay, that's nice, it's a transcriptional change. Are there changes in either protein levels or function that are correlated with changes in the transcript? And in fact, Giulio Agnetti looked at this looked at mitochondrial proteins and mitochondrial proteins using a 2G gel format. These are spots from these 2D gels with a bar plot to the right-hand side of these plots to look at the levels of proteins, of these mitochondrial proteins. Those shown in gray are ones that increased with CRT compared to DHF and increased in a way that seemed to mimic more the control circumstance versus the heart failure circumstance. And what you can see is that proteins in the electron transport chain and pyruvate metabolism, the voltage-dependent anion channel, and a number of other pathways, which are summarized here, energetic pathways, anaplerotic pathways, branched-chain amino acid pathways. So there were over 30 changes in proteins that seem to mimic more closely the control circumstance in cells and tissues that have been isolated from hearts that have been resynchronized. So it's tachypacing resynchronization. So it looked like that even in the presence of persistent tachypacing, with resynchronization alone, what happens is that the energetics seem to resemble more normal control hearts rather than failing hearts. And we looked at that specifically by doing kind of seahorse measurements to look at myocardial oxygen consumption, and ATP generation using a substrate for both complex 1 and complex 2. Partially uncoupled metabolism, so-called S4, was downregulated in DHF and upregulated in CRT. But more importantly, the respiratory control ratio, which is the ratio of state 3 and state 4 respiration, was normalized in the presence of CRT. The ADP oxygen ratio, that is the amount of oxygen consumed per production of a molecule of ATP, also was normalized in CRT compared to control. And the thing that generates the mitochondrial delta Siam or membrane potential, the F1, F0 ATPase was also increased in activity in CRT. So simply resynchronizing tachy contraction actually improved the energetics of the failing heart. So it looks like that heart failure, at least in this circumstance, heart failure is a metabolically driven remodeling that occurs that produces the arrhythmogenic changes that we see in the failing heart. So one more change in gears. We spent a lot of time trying to figure out what might be appropriate targets for treatment of the failing heart to prevent arrhythmias. And we were lucky, this was, again, a long time ago now, to be one of a group of 4 centers that actually got a grant from the Donald W. Reynolds Foundation. Donald W. Reynolds was not the tobacco Reynolds, but he was a guy, and not the aluminum foil Reynolds, but was a guy from Oklahoma who made a lot of money. And he made a lot of money and basically funded a cardiovascular research program. And we were fortunate to get some seed funding to begin this study that we call the Prospective Observational Study of the ICD. It's PROS ICD. It was around the time that Made It 2 was coming out, and we wanted to ask a specific question about what are the features of heart failure or of these patients that would allow for us to better deploy primary prevention defibrillators in this group. We put lots of primary prevention defibrillators in. The question is, how much do they really benefit our patient population? So PROS ICD was originally funded by Donald W. Reynolds, and then we were lucky to get a couple of NIH grants to fund it going forward. So this is the study design. We identify people who have either had prior myocardial infarction and ejection fraction that's low, 30 to 35%, or heart failure that's been present for at least 9 months, like Scud-Heft or like Made It 2. Those people are identified. They undergo some baseline studies, including in about a third of patients an MRI or CT scan. Everybody gets a signal average ECG. Everybody gets blood drawn for genetics and proteomic studies, and everybody gets a clinical evaluation as well as a digital electrocardiogram. ICDs are implanted. They were followed up at least initially every 6 months. And if they had a shock, they came back into clinic as quickly as they could, the earliest convenient time, and we adjudicated the cause of the shock, a separate adjudications committee, redraw blood, digital ECG, and clinical evaluation. In some cases, some patients actually got rescanned. So this allows us to, over time, compare patients in the blue balls here to those who never had a shock to those who did have a shock, represented by the red bar. And again, it's difficult to know who's a case and who's a control. So we had to use a case cohort design, so we compared cases to the entire cohort. And it allowed us to also compare patients who had a shock to themselves over time. So we enrolled about 1,200 patients in this study. I mean, they're reasonably typical, except in Baltimore, what wasn't so typical is that we had about 40% of patients who are African American, which is very unusual for defibrillator studies. Usually those, the diversity in those populations is much less. But they're otherwise reasonably typical, a lot of diabetes, hypertension. They were all pretty sick, ejection fractions in the low 20s. They had class 2 or 3 heart failure symptoms. We excluded those with class 4 heart failure symptoms. And there was a mix of both ischemic and non-ischemic cardiomyopathy. They were pretty well treated for heart failure. Those who are on antiarrhythmic drugs were typically on antiarrhythmic drugs for atrial fibrillation. It's a sick population, and bad things happen to sick people. And despite the fact that everybody got a defibrillator, lots of patients over the first 10 years of the study died. About 300, total deaths were well over 300, but in fact, what was 343? In fact, you would think that it would be easy to adjudicate the cause of death in these patients. No, it's really not very easy to adjudicate the cause of death. We had 40 definitive sudden cardiac deaths that we believe were arrhythmic sudden cardiac deaths. Many of them remain unadjudicated because we don't have the recordings from terminal events, and in fact, somebody's even with a defibrillator is just kind of found dead. It turns out that there were fewer appropriate shocks than there were overall deaths. A number of patients actually went on to heart transplant, mechanical support device, and other therapies. If one looks at the mortality over time, the mortality rate is actually shown in red here, and it's annualized about 5.7%, about 6 years to time to first event. That is about twice the rate of a first appropriate shock. So despite the fact that you get a defibrillator doesn't mean you're immunized from sudden cardiac death or from death overall. In fact, what it does is it reduces the incidence of sudden cardiac death, but certainly doesn't eliminate sudden cardiac death. So this was not surprising to us. I mean, again, this is a sick group. We know we can prevent arrhythmias, but we're not certain. And I think this tells us that and other studies, prospective studies have said that you really can't prevent overall mortality. The interesting thing was we followed these patients throughout the life of their first generator. So these are folks who are now coming back for a generator change, and we asked the question, what's their function look like? And these are people who admittedly were generally well treated in our cohort. But it's very interesting, if one looks at the follow-up ejection fraction after ICD implantation, a full 40% of these people had an improvement in left ventricular ejection fraction with standard therapy. And that's pretty interesting. In fact, there were a number of people in this group, about 25%, who, had they been identified at that point in time, would not have been candidates for a primary prevention defibrillator implant. So merely taking care of folks actually does properly for heart failure, actually improves left ventricular function over time. And in fact, if one looks at, not surprisingly, if one looks at the hazard ratio for all-cause mortality or an appropriate shock, if in fact your ejection fraction improves, the risk of you developing one of those two outcomes actually decreases pretty dramatically. We also took advantage of the fact that 40% of the population was African American. And perhaps not surprisingly, African American patients actually fared worse than non-African American patients. In fact, the all-cause mortality was pretty similar, but the appropriate shock rate was significantly lower in African Americans. So the overall benefit of primary prevention defibrillator in this group, again, it's a prospective observational study, it's not a trial, seems to be a little bit less beneficial. And if one looks at the hazard ratio in pre-specified subgroups of African Americans versus non-African Americans, again, adjusted for age, for gender, for enrollment center, we had basically 3 enrollment centers at the time, Hopkins, Maryland, and the Washington Hospital Center, and other things like the type of cardiomyopathy, atrial fibrillation, et cetera, et cetera. If one looked at all-cause mortality without an appropriate shock, meaning you had a defibrillator, never did anything for you, in quotes. There may have been some backup pacing for sure, but these were all programmed generally in a way where backup pacing rates were really quite low. The hazard ratio for death without an appropriate shock in African Americans was increased by about 33%. So a little bit less benefit in this subgroup of patients for a primary prevention defibrillator in groups of patients who are candidates based on MADIT. It's got to have 4 primary prevention defibrillators. So we wound up genotyping the entire cohort of 1,200 patients on something called the MetaboChip platform, which is about 200 SNPs that have been that include genes that are involved in diabetes, in coronary artery disease, QT genes, et cetera, et cetera. And we were looking for things that have been described to be associated with an increased risk of sudden cardiac death in the population. NOS1AP was one that we described in the Framingham and other studies, BAS2B, a cyclin-dependent kinase and a locus on chromosome 21. Not surprisingly, none of these showed up in this particular genome-wide association, this MetaboChip scan. However, there were a number of others of yet unidentified genes that were associated with, at least in this population, an increased incidence of mortality. Now, this is a terribly underpowered study, 200,000 SNPs and 2,000 patients. So these could be all false positives. But what we've done is we're trying to combine this data set with other data sets to get sufficient power to try and figure out whether or not there are any hits here that are worth going after. So one of the other things that we did was to try and look at other markers that might be associated and help us identify people who are more likely to benefit from a primary prevention defibrillator. We'd drawn blood on everybody, and we'd actually saved both serum and cells. And we were looking at liquid biomarkers to try and figure out if there are any sets of liquid biomarkers that might tell us not only who's at higher risk for dying, but who might be at higher risk for developing an appropriate shock, therefore would benefit from a defibrillator. So we looked at a few different types of markers, CRP and inflammatory biomarkers based on Christine Albert's study and the physician health study, TNF-alpha. Turns out TNF-alpha-2 receptor is more stable over time, so we looked at that, and other myocardial biomarkers. We studied all 1,200 patients, and this particular analysis was based on 137 appropriate first ICD shocks and 343 deaths total. And if one asks the question, were there any clinical markers that identified people who were more likely to get an appropriate shock, the answer was simply no. There wasn't anything that we could identify that identified a patient who was more likely to get a shock. However, there were things that would tell us about patients who are more likely to die despite having the defibrillator in place, and that includes increased age, presence of diabetes was more common in those who died, more advanced heart failure, the use of antiarrhythmic drugs, and if you had a lower GFR. And in fact, in these forest plots, there were a number of things that identified you as somebody who was likely to die despite having the defibrillator in place, and we used the quartile rank of 5 of these markers to develop a score. And this score, basically, quartile 1, 2, 3, or 4, you got a point for each level of quartile you were in for these markers. If you had some of these adverse biomarkers, and this included CRP, IL-6, TNF-alpha, pro-BNP, and TNI, if you had higher levels of these things in aggregate, you were more likely to die despite having the defibrillator in place and not get a therapeutic benefit from the defibrillator. So nothing identified patients at a greater risk for developing an appropriate shock. Therefore, you'd say, got to put a primary prevention defibrillator in this patient to prevent them from dying from an arrhythmia. We then, in the second part of this study, went on to say, okay, liquid biomarkers don't really effectively distinguish people who are likely versus unlikely to develop an appropriate shock. Most of these patients, even after they had the device in place, got an MR, and we looked at late gadolinium enhancement, looked at scar and scar distribution to see whether or not that would help. Despite the fact that everybody had a low ejection fraction, there were patients, about 87 patients, who had no late gadolinium enhancement. Those patients did exceptionally well, regardless of what you did. They didn't need a defibrillator. They had really quite remarkably symptom-free and MACE-free survival. So the second part of this study was to actually ask the question at the time that people were coming in for a generator replacement. And A.V. Kolandelavalu, who's actually a great engineer, developed a number of ways to kind of correct some of these MR scans for the artifact that occurs when you have metal in place. We were looking at the distribution of scar to figure out whether or not that would help us decide whether or not a patient really needed a defibrillator or would benefit from a defibrillator. So we looked at regional function. We also looked at scar and scar distribution. Let's see if I can do that. Scar and scar distribution in these patients. And that study right now is ongoing. We're kind of in the middle of it. We've got about 400 people who we've rescanned, and we're trying to sort out whether or not the distribution of scar, the amount of scar, predicts who in the second generation of device, their second generator, is likely to have a firing for VT or VF. The one other thing we've done, taken advantage of in imaging this group of patients, is we've worked with the biomedical engineers at Hopkins, particularly Natalia Trianova. One of the things that's actually most predictive and that we use in the EP lab is the induction of arrhythmias with programmed stimulation. So it's an invasive way to do things. You'd like to be able to kind of do this in a way that perhaps you could do minimally invasively. So what Natalia and Kathy Wu did was to create from MR a detailed model of the left ventricle. And the detailed model of the left ventricle included scar and scar distribution, included fiber orientation, and they overlaid areas of scar, and the scar shown here in yellow and green. In green is the so-called gray zone, which is a mixture of normal and scarred myocardium. The yellow is dense scar. So Talia created a model of the left ventricle and then did simulated program stimulation using a computer algorithm, and that's shown here, and paced from a number of different spots, and used a typical extra stimulus protocol. And what you can see is that in some cases let me go back to the beginning. Pace 1, pace 2, pace 3, and then induction of ventricular tachycardia in this particular heart. And what we tried to do is correlate the simulated induction of VT with actual events in these patients. And in fact, there was a reasonable correlation between people who had appropriate shocks from their defibrillator for VT and VF and the simulated induction of ventricular tachycardia using completely in silico cardiac model and in silico program stimulation. Don't worry, this is very intense to do, so the clinical electrophysiologist in the audience will still have a job. You'll still need to do some program stimulation. By the way, nobody does this anymore. You guys spend all your time doing AFib ablation, so no need to worry. I think what I'd like to do is summarize here. We spent a lot of time trying to figure out what the fundamental changes are in the electrophysiology of the heart that might predispose to the development of arrhythmias and result in sudden cardiac death in patients with heart failure. There are a number of electrical remodeling changes, including reduction in potassium currents, changes in calcium handling, reduction or alterations in network function of the heart, all of which are targets, albeit they're targets that have been targeted therapeutically without substantial benefit. It is a metabolically driven process, and perhaps we need to go further upstream, in fact, address metabolism. There is regional remodeling of both structure and function. I think CRT is primarily a metabolic therapy. It's a metabolic therapy that improves energetics of the heart, and I think does so, and in doing so, improves outcome. I think there are further to be identified genetic, genomic, and protein biomarkers, as well as imaging biomarkers that might help us better figure out who's most likely to benefit from an ICD. The most important part of the slide are the folks who, mostly at Hopkins, but at the University of Maryland, University of Utah, and others who've helped with this work. And I'm delighted to be here. Happy to take any questions that you might have about any of this. So thank you very much for your attention.